Hi! After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example like eye color. Yes, your DNA has a genetic information that codes for the color of your eyes. Your eye color is based on the pigment that is inside the eyes. But in order to have that pigment, you have genes, which are portions of DNA that can code for proteins which help make that pigment. So what we're to talk about is about your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means make something. So protein synthesis means to make protein. What's the big deal about proteins? Proteins are a kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. Proteins is essential for you to live. Protein synthesis is the process where a sequence of DNA is used to build a protein from individual amino acids. The two processes or steps in protein synthesis are transcription and translation. It's a transfer of information from DNA to RNA to proteins or gene expression. Let's try to answer this one. Let's complete the diagram and identify the amino acid sequence coded by each mRNA. The given DNA strand are A, T, G, G, T, A, G, C, T, A, C, C, T, and T. First, we have to fill in the complementary strand. Remember, T-thymine pair with A adenine and G-guanine pair with C cytosine. So here, the complementary DNA strand is T, A, C, C, A, T, C, G, A, T, T, G, G, A, and A. The first step in the process is called transcription, where a coding region of DNA is converted to messenger RNA or mRNA, the first type of RNA. Here, it transcribes or copies DNA's message or the genetic code in nucleus. Part of the DNA temporarily unzips and is used as a template to assemble complementary nucleotides into mRNA. During transcription, mRNA is made from the DNA sequence, following the base pair rule in RNA. Uracil pair with adenine and guanine pair with cytosine. For the second procedure, we have to fill in the correct mRNA bases by transcribing the bottom DNA code. So here, the mRNA bases are A, U, G, G, U, A, G, C, U, A, C, C, U, and U. The second step or process in protein synthesis is translation. This is the process of converting or decoding the information in mRNA into a sequence of amino acids to make a protein. In translation, the mRNA leaves the nucleus and goes to a ribosome in the cell cytoplasm. The ribosome reads the message 3 bases at a time. The 3 bases of mRNA is called codon. It will code for 1 amino acid. The amino acids are joined together and folded into a protein. Transfer RNA or tRNA, the second type of RNA, carries amino acid from the cytoplasm to the ribosome. tRNAs join and bring correct amino acids. Each tRNA has 3 nucleotides that are complementary to the codon in mRNA. The 2 parts of tRNA are anticodon and amino acid attachment site. tRNAs move from A site to P site. Then the A site is now open for new with attached amino acid to join. For the third procedure, we have to fill in the correct tRNA bases. So we have U, A, C, C, A, U, C, G, A, U, U, G, G, A, A. The third type of RNA is the ribosomal RNA or rRNA. Allows amino acid to attach by making peptide bonds or to assemble protein. The ribosome recognizes a stop codon. The polypeptide is terminated and released. For the fourth procedure, translate the mRNA codons to find the correct amino acids. We will use the genetic code. The genetic code is the language in which instructions for proteins are written in the base sequences. It is used to decode mRNA message. So here, the first letter is A, second letter is U, and the third letter is G, and that is methionine. The G, the U, and the A is valine. The G, C, and U is alanine. A, C, asparagine, and the C, U is the leucine. So the amino acids here is methionine, valine, alanine, asparagine, and leucine.